everyone for the CAFNA Research Council seminar today. I'm obviously not Dr. Jose, but I'm representing CRC as Animal Sciences faculty member representative on the council. So I'm here to introduce and take questions for Dr. John Driver, who's today's speaker. Driver earned his bachelor's and master's degree from the University of Pretoria in South Africa, and then his PhD in animal nutrition from the University of Georgia. After that, he studied the genetics of type 1 diabetes at the Jackson Lab in Bar Harbor, Maine. He's coming to us actually in January of this year as an associate professor here in the Division of Animal Sciences. And he's coming from the University of Florida, where he started in 2012 and became an associate professor in 2019. While there, he served as the director of the Animal Molecular and Cellular Biology graduate program and really got his research going in two distinct areas, which we'll see today. One is the immune system of pigs with a focus on the contribution of innate T cells to immunity. And the second is pathogens that are important for swine and human health. So I think Dr. Driver is going to get us started here today. And again, we thank him for presenting and you all for being here.
All right, so I hope you can all see my presentation, and thank you for that introduction, Alison. Just get myself sorted out. So I'm very pleased to be here at the University of Missouri, now part of the CAFNA family. As Alison mentioned, we're a swine immunology lab, and my program uses swine models for the dual purpose of understanding pig and human immunity. So because this is an immunology talk, I thought I'd start out with a quick refresher on the components of the vertebrate immune system. So there are two main branches represented by these two big circles. The first is the innate immune system, and this is the first to respond to an initial infection. And it does not retain memory from previous responses. It consists of a lot of big cells like macrophages and dendritic cells, a lot of granulocytes, and we also have some lymphocyte members like NK cells. And the innate immune system is just a mere 600 million years old. Then if a pathogen persists, despite the innate immune defenses, you have the adaptive immune system. And the adaptive immune system is highly specific to particular pathogen and can provide long-lasting immunity, and it consists of B cells and T cells, and it's just a mere 450 million years old. And these days we think of another kind of immune system we call the bridging immune system over here, where we have the overlap. And these are B cells and T cells biologically, but they lack the pathogen specificity of conventional B cells and T cells. Instead, they recognize pathogens in a very generic way, similar to the innate immune system, which is why we call them innate adaptive immunity. So my talk today is going to be focused on the T cell members of this bridging immune system. So these bridging T cells, they develop in the thymus, which is this gland above the heart, like all other T cells. I've got a flow cytometry plot here on the left-hand side, which you'll find in most basic immunology textbooks. We call it the airplane plot. And it shows the basic development of T cells that come from the bone marrow. So they come to the thymus. At first, they're negative for CD4 and CD8 molecules. Then they acquire both, and then they lose one or the other as they become mature T cells and they leave the thymus. So this view of T cell development is founded on the major histocompatibility complex, or MHC system. And these MHC molecules present bits of pathogens to T cells in the immune system to generate an immune response. So this view of T cell development is actually quite a gross oversimplification, because it doesn't represent the tremendous diversity of weird and wonderful minor T cell populations which also develop in the thymus. And although some of them are extremely rare, they can have powerful and profound effects on immune responses. But we're now gaining a much better appreciation of this diversity through new technologies like single-cell sequencing, whereby we can characterize the transcriptome of single cells and then cluster cells based on similarities and differences in gene expression. So this is a figure from a recent paper that visualized the cellular composition of the human thymus, and they've integrated fetal, postnatal, pre-adolescent thymi data sets capturing the whole period of thymic function. So here are those double negative thymocytes, and then here are the double positive thymocytes. And over here we have all the mature T cell types. Out here we have gamma delta T cells, which are a little bit different. And here's the CD4s and here's the CD8s. But what I want to draw your attention to is the diversity of T cells that you get in the thymus beyond CD4 and CD8. Even this is a gross oversimplification though, because you can, for instance, drill down into just the gamma delta T cells, and there are half dozen different subsets of gamma delta T cells. They have unique receptors, cytokine profiles, effector functions. They move through to different tissues. They've got different jobs to do. Beyond that, there's some T cells in the thymus that are so rare that's difficult to detect them even in this galaxy of single cells from thymus data. And really to study them, you need reagents to purify and concentrate them first. So it's in some of these T cells, these very rare T cells that I'm interested. So we call them unconventional T cells. These are T cells that recognize unconventional antigen presenting molecules that are different from MHC molecules. And these antigen presenting molecules here are highly non-polymorphic molecules, meaning they don't vary in their sequence from individual to individual and even among species. Some of them are CD1A, B, C, and D. You also get HLA-E and another presenting molecules, MR1. So T cells that recognize these strange antigen presenting molecules are called unconventional T cells for several reasons. One of them is they don't recognize peptide antigens like most other T cells. And secondly, these cells circulate in a fully differentiated state, poised to trigger immune responses at a moment's notice. Another really interesting aspect of these cells is their tremendous species diversity in types and subtypes and functions. Just one example is, humans, we have T cells that recognize antigen presenting molecules A, B, C, and D, but mice, they just have CD1D. They don't have CD1A through C, but they have a lot more T cells that recognize CD1D than humans do. So what do these unconventional T cells recognize? This has actually been a difficult question to answer, but what we're realizing now is that these CD1 molecules present different kinds of lipids and glycolipid molecules, many of them from the cell walls of microorganisms. And all the different CD1 molecules have different expression patterns in the body and probably playing different roles in immune responses. And then you have cells called MAIT cells, and they recognize MR1. And MR1 presents derivatives of vitamin B9 or folate to T cells. And this seems to be important for controlling microorganisms that utilize the riboflavin pathway. Nowadays, we have synthetic ligands for these antigen presenting molecules. And that's really handy because now we can isolate them and stimulate them. We're starting to get a much better understanding of what they're doing in the immune system as a result. So an important aspect of these unconventional T cells is how they respond to stimulation. So for conventional CD4 and CD8 T cells, the cost of maintaining a diverse, naive T cell pool that can recognize almost anything out there is that any given pathogen-specific T cell precursor must be very rare at the beginning. and unable to provide immediate protection. And then what happens is, upon encounter with the pathogen, activation of these rare cells drives clonal expansion and generates a large effector T cell population. And then eventually, upon re-exposure, you have memory T cells, and they can respond quickly and strongly. Just like when you get your booster shot from your COVID vaccine, the second shot you react to much quicker and much bigger. In contrast to your conventional T cells, unconventional T cells, they already are circulating in a kind of memory state, and they're already poised for rapid responses, their effect at this stage. As a result, the way they respond to an initial stimulation is the same way they respond to a secondary stimulation. There's no real difference there. So to summarize, these unconventional T cells can be thought of as part of the modern adaptive immune system, which has evolved to take on characteristics of the more ancient innate immune system. And we think their goal is likely to augment the innate immune system during early stages of infection. But they can also shape adaptive immune responses, B cell responses and T cell responses. So where exactly do these cells fit into an immune response? Now I focus on MAIT cells and NKT cells as we know the most about these. So they're both important regulators of host environment interactions, and they concentrate themselves in barrier organs like the airway and the skin and the gut. And when there, they can be activated by ligands presented by the antigen presenting molecules. But they also express a large array of cytokine receptors. So receptors that can recognize lots of different kinds of immune molecules. And they can be activated independently of their antigen presenting molecules. And when they do, these cell types are particularly talented at producing large amounts of many different kinds of immune molecules very rapidly that do things like recruit components of the innate immune system, like your granulocytes and mast cells. They can also skew the development of local adaptive immune cells like T cells, and they can induce B cells to start making more and different kinds of antibodies. So these responses in barrier organs modulate a host of different diseases. We know these cells are really important in asthma, COPD, atopic dermatitis, psoriasis, as well as many infectious diseases in barrier organs. So we know NKT cells and MAIT cells make up a substantial component of immune responses to microbial infections because of studies like these two that I'm showing you here, where they infected mice that are deficient in MAIT cells because they don't have MR1, or NKT cells because they don't have CD1D, and they're using influenza as the pathogen here. And without MAIT cells, mice succumb to influenza much more rapidly. Same thing for the NKT cell deficient mice. And you can see if mice don't have NKT cells, they produce much higher levels of virus in their lungs. So we've now thrown, the community has thrown all sorts of pathogens at these knockout mice. For instance, we now know that NKT cell deficient mice are more susceptible to a host of different kinds of microbes, ranging from viruses all the way over to helminths. Our lab loves and studies type 1 NKT cells. So these are T cells that are restricted to the CD1D molecule. And they're also called invariant NKT cells because they have a highly conserved T cell receptor, meaning that their little receptor here, the alpha chain and the beta chain, they're exactly the same on every NKT cell. And they're going to be the main focus for the rest of my talk. So a really compelling reason to study these cells is that in mice and in humans, NKT cells react with this very particular glycolipid antigen or molecule called alpha-galactosyl ceramide. Here it is. So about 30 years ago now, this molecule was isolated from a marine sponge by a company searching for products with antimetastatic activities. And when this molecule is presented by the CD1D molecule, it strongly activates NKT cells and gets them to produce a large variety of cytokines simultaneously, both pro-inflammatory and anti-inflammatory cytokines. So this means that in theory, you can use NKT cells to activate, oh, sorry, skipped a step here. Since then, they've made several synthetic derivatives of alpha-galactosyl-ceramide, and you can use these to activate NKT cells and promote them to make more pro-inflammatory cytokines or more anti-inflammatory cytokines. And the nice thing about that is that you can tailor-make your immune response depending on the type of disease you're trying to treat. So since we've discovered alpha-galsa, there's been a large number of preclinical studies, mostly in mice, performed to investigate whether NKT cell-based therapies can be used to treat infections, cancer, autoimmune diseases. A lot of these studies have looked into whether you can harness NKT cells to adjuvant vaccines as well. So when I joined University of Florida as an assistant professor, I became interested in whether NKT cells could be targeted in livestock to improve animal health, but also wanted to ask whether we could use large animals as a translational model to evaluate the safety and efficacy of NKT cell therapy for humans. So a major advantage of targeting these NKT cells is that you can use the same alpha-galsa molecule across species. So you don't need to make a mouse version of alpha-galsa and a pig version and a human version, because that CD1D molecule is so non-polymorphic. One alpha-galsa fits them all. And the species I chose to work in was pigs. So why pigs? Well, they're susceptible to many human pathogens, more than 40 and counting. And so we could ask questions like whether we can activate NKT cells to treat infectious diseases that are important for pigs. They are an important source of zoonotic diseases. So keeping pigs healthy is keeping humans healthy. They're of comparable size, anatomy, development, and metabolism to humans. So they're great for dosage and safety studies. Unlike mice, they have other CD1 molecules. So you can study NKT cells in the context of all these other CD1 restricted T cells. And they're a critical livestock species in need of novel solutions for existing and emerging health challenges. Another really good reason to study the system in pigs is that pigs actually have an NKT CD1D system. So as I mentioned earlier in my talk, there's tremendous species variation in these unconventional T cell types. And for instance, you couldn't study the system in ruminants. They don't have a system that's the same as the system you find in mice and in pigs in humans. And lots of other species we're just not sure about at this stage. So we've, in the last few years, characterized these cells in pigs. One of the things we're particularly interested in now is identifying subsets of pig NKT cells. So mice have taught us that NKT cells fully differentiate in the thymus into separate lineages of NKT cells. So here we have NKT1 and NKT2 and NKT17. And these subsets have their own transcription factors, and they have their own capacity to produce different types of cytokines. And they come out of the thymus already fully differentiated into these subsets, which is different from conventional CD4 T cells, where Th1, Th2, and Th17 T cells must differentiate after stimulation in the periphery. These different subsets of NKT cells home to different tissues And in those different tissues, they have their unique functions to fulfill, and they play unique roles in disease. So we wanted to find out if pigs were the same. The problem is that we just didn't have the reagents to be able to do this in pigs, so we took a single cell sequencing approach. So after a very difficult purification process, because these cells are so extremely rare in the thymus, we were able to get 12,000 of these pig NKT cells, and here they are. And then we combined our data with the published single-cell-seq NKT dataset from 9-week-old C57 black 6 mice. So in the mouse dataset, we saw everything we expected. So here are those NKT1 cells, and here the NKT2s and the NKT17s. They all have their own transcription factors that they upregulated. When we looked at pigs, they all resemble mouse NKT2 subsets. So mouse NKT subsets look completely different from pigs. And pigs seem to be much more immature than mice, because NKT17 and NKT1 subsets are derived from NKT2 cells. So because of this big difference, we think that this has important implications for how mouse and pig NKT subsets contribute to immune responses. So we've accumulated a lot of information about pig NKT cells, but what we really want to know is whether NKT cells make important contributions to immune responses in pigs. So we had to choose a disease model, and we chose the swine influenza challenge model because it's a very reliable translational model of human influenza infections. Pigs are natural hosts of flu viruses, whereas mice are not. Also, pigs, they give rise to new influenza strains that occasionally give rise to human pandemics, since they can be infected with, avian strains and human strains. They got their own strains, and sometimes they recombine. So establishing that NKT cells make important contributions to swine influenza immunity has important implications for human and pig health. So we've asked many questions. I'm just going to touch on 3 of them today here. We wanted to know, are NKT cell knockout pigs more susceptible to influenza infection like we see in mice? We also wanted to know, could we use an NKT cell agonist like that alpha-galsa molecule I mentioned to you to increase the efficacy of an inactivated killed influenza vaccine like the regular flu jab we get, which are not that good at providing protection against viruses that are not in the vaccine. And we wanted to find out whether NKT cells are involved in a very dangerous pathology called vaccine-associated enhanced respiratory disease. So to the first question, to test the contribution of pig NKT cells to influenza immunity. So when I started off as an assistant professor at University of Florida a few years back, I applied to the National Swine Research and Resource Center here at University of Missouri to create me a CD1D knockout pig. And I was really delighted when they accepted my application, and they generated seven founders with distinct CD1D disruptions, and none of those pigs had NKT cells. So they must know what they're doing over here. And then we used these pigs in a study where we infected them with pandemic H1N1 influenza virus, along with their littermate controls. So the green pigs are the knockouts, don't have NKT cells. And then we had littermate controls that do have NKT cells. And then we also have pigs that have NKT cells, but we didn't infect them as controls. So we found that virus shedding was significantly delayed and reduced in the knockout pigs. And we found that some of these knockout pigs never ended up shedding virus. Additionally, we found that knockout pigs had lower levels of virus in their lung lavage fluid, bronchus, and nasal turbinates. So the interesting thing is our data directly contradicts the several mouse studies that are out there showing that influenza infections in CD1D knockout mice are considerably worse than in CD1D intact mice. So you can see the CD1D knockout mice produce a lot more virus than CD1D intact mice. We see the opposite. So these data indicate that in pigs, NKT cells appear not to be involved in controlling influenza virus replication, but rather they promote it. And this disparity may be due to several factors, including the difference I mentioned between mouse and pig NKT cell subsets, as well as that mice and pigs have considerable differences in the proteins they express that are involved in antiviral immune defenses. Another reason is that mouse strains used for flu studies and NKT cell studies usually develop much more severe respiratory disease after influenza infection compared to pigs due to these differences in antiviral immune defenses. So because pigs are immunologically more similar to humans than mice, we think it's likely that our knockout swine model more accurately reflects the contribution of NKT cells to flu immunity than the NKT deficient mouse model. So on to the next topic I want to talk about, and that's testing whether NKT cell agonists like alpha-galsa can be used as an adjuvant for inactivated influenza vaccines. So adjuvants are substances that we use to boost the scale, the scope, or the quality of an immune response elicited by a vaccine. So the problem with inactivated flu vaccines, like the common flu jab we all get, is that they're usually pretty ineffective at generating cross-protective immunity against viruses that are not in the actual vaccine cocktail. So we wanted to find out if vaccination with alpha-galsa as an adjuvant would improve this cross-protection, as there's considerable evidence in the mouse literature that it does. However, to start out with, we asked whether this concept would work in a much simpler homologous virus challenge system. So a homologous challenge system means that the virus that you infect the pigs with is the same virus that you put in the vaccine. So this is relatively low-hanging fruit, and we always expect to get pretty good protection. Here are our treatment groups. So we have pigs that were mock vaccinated and mock challenged, negative controls, and then challenged, but without any vaccine, positive control. And then the purple pigs, these are pigs that were vaccinated with pandemic H1N1 virus and challenged with the same virus. And then the fourth group is just like the third group, except that it was adjuvanted with alpha-galsa. And the pigs got 2 vaccines, 16 days apart, and then 16 days later they were challenged. And you can see it's only the pigs that were vaccinated with alpha-galsa that have a big increase in their circulating levels of NKT cells, indicating that they were indeed activated. So with the addition of alpha-galsa, we got everything we had hoped for. Much bigger boost in virus-specific antibodies and virus-specific T cells in the lungs and in the periphery, which are good indicators of protection. And indeed, that's what we found. So these alpha-galsa adjuvanted pigs, they shed a lot less virus than pigs that just got the vaccine alone. And this is in log scale, so the difference is actually large. And then we were simply unable to find a virus at 3 and 7 days post-infection in adjuvanted pigs, whereas we could still detect virus in pigs that received the vaccine alone. So these results demonstrate that alpha-galsa can boost vaccine-mediated protection against a homologous influenza infection. That's all good and well, but the real challenge with inactivated flu virus vaccines is inducing cross-protection, as I mentioned, against non-vaccine-associated strains. So, of course, the more distantly related the viruses are between infection and vaccine, the more difficult it is to induce protection. So next, we designed a study to determine whether alpha-galsa alone or in combination with an oil and water adjuvant, ISA 25, would improve protection against a heterologous virus challenge. So a heterologous challenge means that you infect with a virus of the same HN subtype as the vaccine, but the viruses come from different clusters. They're a little removed. Okay, so here are different treatment groups. So we have vaccine, and then vaccine with the commercial adjuvant, which we wanted to compare against vaccine with alpha-galsa. And then we put both kinds of adjuvants together, the ISA 25 and alpha-galsa. And then we have the 7th treatment group, where in the first vaccination, they got ISA 25 as the adjuvant, and in the second vaccination, they got our alpha-galsa to activate NKT cells. And then to test heterologous immunity, we subsequently challenged the pigs with Colorado 99, which is another H3N2 virus, but belonging to a different cluster than our Texas 98 H3N2 virus vaccine. So this slide shows the virus titers in lung lavage fluid when pigs were necropsied at 5 days post-infection. And it illustrates well the challenge of inducing cross-protective immune responses, even against a relatively closely related virus. So here are the pigs that were infected with no vaccine. And you can see the yellow pigs did get the vaccine, but not much of an improvement here. And then green, these are pigs that got the adjuvant with the vaccine, the oil and water adjuvant. Here's alpha-galsa, didn't do much better than the oil and water. The combination for some reason did worse than either alone. And here we have the one, then the other. The bottom line is our vaccine formulations, including our alpha-galsa adjuvant, did not produce really meaningful cross protection compared to pigs that received no vaccine at all, which is rather disappointing. You know, we're hoping that alpha-galsa would tip the scales and we get good cross protection. Although we didn't get this, we did find an interesting observation in the study. And we've repeated it, we find the same thing. And that is that when you use alpha-galsa as an adjuvant, especially alone, we tend to get actually worse lung disease than we would if we didn't give any vaccine at all. And we've looked into it, and it seems to be associated with an accumulation of virus-specific T cells in the lungs of pigs that receive alpha-galsa. So that's what we're looking at here. And these virus-specific T cells produce large amounts of an inflammatory molecule called interferon gamma. And it's this molecule that drives lung inflammation. So we have lots of these inflammatory cells when we use this molecule as an adjuvant. And this result got us thinking whether NKT cells could trigger a dangerous phenomenon known as vaccine-associated enhanced respiratory disease, or VAERD for short. So VAERD is a disease characterized by the development of very severe lung disease, and it arises with certain combinations of vaccine and challenge viruses. And it results in lung disease that's much worse than if you had been infected and never had a vaccine at all. And it's a condition that develops only with the use of inactivated influenza vaccines, not live attenuated. And you need a virus of the same hemagglutinin subtype or HA subtype as the infection strain, but with substantial evolutionary divergence. So a consistent feature of this disease is the presence of vaccine-induced antibodies that will cross-react with the infection or challenge virus, but these antibodies fail to neutralize the virus. Instead, they tether the virus to your cells, and in that way actually enhance the ability of the virus to infect the cell, and that leads to worse lung disease. So this type of enhancement, of course, is a major concern for vaccine developers, because although it's rare, when it does happen, it's very dangerous. Because of our previous results, we wanted to find out if NKT cell agonists used as adjuvants could trigger this very dangerous phenomenon called VAERD. So this is a study we performed, another vaccine study. In this study, pigs were vaccinated 3 weeks apart. And this time we used an H1N2 virus, Minnesota 08. And this was adjuvanted with an oil and water adjuvant over here. commercial oil and water adjuvant, or we adjuvanted with our alpha-galsa, or the pigs got a combination of oil and water and alpha-galsa. And then we challenged the pigs with a heterologous virus. Again, we used the pandemic H1N1 virus, strain California 04. We don't have a treatment group without oil and water because an adjuvant is absolutely critical for the development of VAERD. Okay, so what did we find? We found that pigs vaccinated with alpha-galsa, with or without the oil and water adjuvant, had extremely severe clinical signs and lung pathology compared to pigs that were unvaccinated. And we found that clinical signs were particularly severe in both treatment groups that received alpha-galsa. In fact, some of these pigs didn't make it to the end of the study, and they had to be euthanized early. So it's a very interesting result to us. We're able to trigger VAERD using very tiny concentrations of a very tiny molecule targeting an extremely rare type of immune cell in the immune system of pigs, these NKT cells. And this is quite different to previous models of VAERD, which use these oil and water adjuvants that stimulate a very broad immune response. We've looked for potential mechanisms of action. Straight away, we examined concentrations of antibodies against the hemagglutinin stalk protein, as these are thought to drive VAERD because they attach to the challenge virus and attach the challenge virus to the cell they're going to infect. But surprisingly, pigs vaccinated with alpha-galsa alone had lower concentrations of these stalk antibodies compared to pigs that got the oil and water adjuvant, even though they had just as much lung disease, if not more. And then next, we looked at concentrations of virus-specific T cells and found that alpha-galsa adjuvanted pigs, they had just tremendous numbers of vaccine-specific T cells and virus challenge-specific T cells in their lungs, producing inflammatory interferon gamma, reminiscent of the last vaccine study I showed you. So we think it's this accumulation of lots of inflammatory T cells into the lungs that are driving VAERD with NKT cell therapy. And if so, this represents a really novel mechanism through which VAERD is stimulated. So in conclusion, VAERD can be triggered by stimuli originating from very rare NKT cells. NKT cells appear to induce VAERD via T cells rather than these tethering antibodies. And our findings are, I think, important because currently most efforts to translate NKT cell therapy to humans are focused on using alpha-galsa molecules as vaccine adjuvants. There's a tremendous number of studies going on about this. So there could be important safety concerns with this approach. So that concludes my talk, except to say that now that I'm here at University of Missouri, we're making new and exciting pig models to determine the role of other kinds of innate immune cells in swine immunity to influenza and other pathogens. So I'm working with my friends over in animal sciences, Dr. Kehoe Lee and the rest of the NSRC crew, to make pigs that lack these other kinds of cells. And with that, I'll leave it there and take any questions.
Thank you so much for that great talk. So we're going to take questions in the Q&A function. So if you have any questions, please type those in and we'll read those. So while we wait on the first questions to come in, I'll say that I'm always taken by the tremendous variation in responses to vaccine. Do you think that the NKT cells are involved in that? What, am I seeing more variation or am I more surprised by it than I should be by some of your data or some of the responses, I should say?
Well, yeah, there are lots of different factors, of course, in a person's that will determine a person's efficiency at producing a vaccine response. Of course, a big one is the MHC molecules that they express, which will present different parts of the vaccine to the immune system. But to your point, yes, I do think a lot of this variation is coming from these innate type T cells, because much more than your conventional type of T cells, among individuals, there's just massive variation in the frequency of these cells. So among human beings, for instance, there's more than 100-fold difference in the frequency of NKT cells among individuals. And then, of course, that's just one type of innate immune cell. I wonder if I can just show you an example of this. So I have a slide back here. So this is, we analyzed the frequency of NKT cells in litters of 15 different sows. So each dot is a piglet. And so, you know, in some piglets, 2% of their T cells were NKT cells. And then we have plenty of litters where you barely detect an NKT cell. And it's the same thing with humans. And that's correlated with a whole bunch of diseases. It also represents a big hurdle for harnessing these cells. And that's what they're finding in the human therapy field. Because, you know, one dose is definitely not going to fit all. You're going to get a wonderful response in some people when you target these cells, because you get a lot of them, and then almost nothing in others. And that's going to be a problem when trying to harness them as adjuvants for vaccines.
Great. So Rod Geiser is asking, does the increase in IN or IFNG have an effect on protecting the virus or infected cells from being targeted?
Yeah, so interferon gamma is, though, you know, what everyone wants to induce when you have a virus infection, because it's just a really important arm of antiviral immunity. But like a lot of things in the immune system, it's a real double-edged sword. You know, like we're finding with COVID, you want a nice, robust immune response, but then half the time that's what kills you as well. So you have to have things in balance. In this case, you can see that with our VAERD model, we get lots of interferon gamma producing cells in the lungs. And when we looked at virus concentrations in the lungs, well, we didn't substantially reduce it compared to pigs that were vaccinated, that were infected without any vaccine at all. So it probably induces a lot of immune cells come into the lung, and they all get in the way of each other, and it's difficult to resolve the infection for that reason. So yeah, this is a great question, and we're always thinking about as immunologists.
So we don't have any other questions, well, at the moment, except that Dr. Geiser did respond that the pig conceptus does produce a lot of interferon gamma, as he is prone to point out. So if you have anything to add in how your work maybe helps explain that.
Well, I'm not sure whether our cells are, how they contribute to that, but yeah, that's a good point. Yeah.
So in that variation that you were talking about, what do you think is really causing that much variation, especially within the sows and their litters? As someone who works in how, you know, pregnancy affects animals for the rest of their lives, do you think that is more, you know, based on the sow, based on what happens to her during pregnancy, based on her environment, her previous, you know, disease challenges? What's causing that?
Yeah, definitely an interaction there, but primarily genetics. So, you know, I come from the mouse world, and I got into NKT cells in mice, and we worked out the genetic locations of genes that influence whether a mouse strain is going to have very high or low levels of these cells, and the different subset ratios and everything like that. So what we're seeing in that slide I showed you is probably primarily genetics driven. Yeah, so you know, in mice, you can make a beautiful, we did this once, a beautiful ladder showing 40 different mouse strains, ranging from strains that have just huge numbers of these cells to undetectable levels of these cells. That said, there are environmental factors that can stimulate NKT cell proliferation. Vitamin D was a hot one for a while. So they showed that without vitamin D, early in life, you never really develop many NKT cells. You have to have them for thymic development of NKT cells. And certain microbes can stimulate NKT cells to expand in the body, and that persists for a long time. So a combination.
Great. Well, any last questions? I'll give it another minute or two. Again, in the question and answer or the chat, either one. You can see both. And Rhonda, would you like to put up the slide for the next one while we're waiting to see if there are any other questions? Great. So a reminder, while you're thinking of your last questions, that our next CAFNA Research Council webinar will be on Thursday, April 7th. So we're skipping next week, of course, because of spring break. And Dr. Dillon, who's an assistant professor in food, nutrition, and exercise science, is going to be talking with us about cardiometabolic health and some host microbiome interactions. So that should be a great talk again on Thursday, April 7th at 3:30, webinar only. Well, seeing that no other questions came in, I'm sure that Dr. Driver would be happy to answer them by email or phone call or dropping by his office. So let's virtually thank him for a great talk. And again, thank you all for attending today's CRC seminar.
Thank you all.